Hello, and welcome to another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteris Associates. Today I'm going to talk about hepatitis C for the third time. In the first one, I talked about the discovery of hepatitis C at Chiron Corporation and the group led by Michael Houghton. Then number two, I talked a bit about variation in hepatitis C, particularly with regard to genotypes and the impact of that on development of drugs. Talked a little bit about culture as well, which was given to Charlie Rice, one of the other recipients of the Nobel Prize in 2020. That led to the development of antiviral therapies, but that didn't happen until the mid-2000s. So I'm going to go back in this episode to before that. In fact, back to the 1990s again, just after the discovery of hepatitis C and the process that we went through, the progress that was made in understanding much more about the virus, much more about how to measure it, and much more about how to treat it. So way back in episode 7 of Interesting Stories, we talked a bit about HIV viral load. Today's going to be hepatitis C viral load, but a lot of the same things are going to be pertinent here. The importance of understanding the viral load itself, how much virus there is in a particular sample, and in particular, we're going to focus on blood. The predictive value of that. For instance, in the case of HIV, the amount of RNA in someone's blood was a predictor of developing AIDS. And then the use of it in understanding the application of therapeutics. So that means then looking at the level of virus change, hopefully down if you treat someone, and that being linked to the actual clinical outcome. So those are referred to as clinical surrogate endpoints, which are quite valuable. It was in HIV, and as you'll see, it was in HCV as well. So as I described briefly in the case of HIV viral load, we developed a technology at Chiron Corporation called branched DNA signal amplification, or bDNA. We used that same approach for the development of HCV viral load testing. We originally developed the test in our clinical reference laboratory, so it was available only in that spot. So people had to send us samples from all over to actually perform particular studies. Very early on, there wasn't any significant amount of treatment available. There weren't any typical antivirals at all, mainly because of the difficulty in culturing the virus to be able to use that. There was an immunotreatment that I'll come back to in a little while, but this was the early days. And we now for the first time had a tool where we could determine how much virus is actually in the blood. So there was a lot of interest in this. I mean, we spent a fair amount of time going to talk to thought leaders. That was led by our commercial team that was headed by Mike Ritchie, who we've talked about before. And we started out with things such as liver transplants. Terry Wright at UC San Francisco was our collaborator. She was a major figure in liver transplant. What had happened was a number of people received liver transplants from people that were infected with HCV, but they didn't know it. I mean, nobody knew it because there was no way to test it. So we looked at individuals that Terry had cared for that had transplanted livers to determine whether or not they had HCV infection, and a number of them did. So we're able to use this as a monitor of that. As I mentioned in the last episode, of course, we had developed at Chiron, with collaboration of others, immunoassays to look for antibodies in individuals. But in the case of the liver transplant, those people are immune compromised, and there were very low levels of antibody if you could find them at all. So RNA testing became really very critical. As this process to engage the thought leaders went on, I brought into the whole team here my colleagues Judy Wilbur and Janice Kohlberg and others as well. But the three of us did a lot of travel to meetings all over the world, meeting with gastroenterologists. And they all had something they wanted to study and wanted to engage us in doing so. And we took on almost all comers. So we did a lot of work in this area for quite a while. It was very interesting because in the United States, the gastroenterology community has these people who are local experts in places like Los Angeles or Miami. And there'll be a particular thought leader there who was the key person people went to for these hepatology issues. For instance, a woman named Karen Lindsay in Los Angeles, who was the person. So we would go to those people and talk to them and develop studies. I go to a place like Italy, and as opposed to there being just maybe 2 or 3 individuals you need to talk to, as we had in France, there were like 60. I mean, everybody was an expert in their area, and it was very amusing to go to some of their meetings and see the knockdown, drag out fights they got into with one another about the whole field. But I couldn't understand them, so I didn't know what was going on. And they were always nice to us afterwards, so we didn't have a problem with it. So the types of studies that people proposed and we helped them to work on were things such as what's the variation in viral load in one person versus another at different stages of disease, chronic individuals versus acute infections, co-infections with HIV or hepatitis B, or what later was discovered, the virus hepatitis G, which ended up not causing disease, but it was present in a lot of people and had an impact on the amount of HCV viral load. It tended to be higher in those people that were infected. We looked at people that were hemophiliacs, people that were under dialysis, people that had received immunoglobulin immune treatment for a variety of different diseases because there was concern about there being contamination in those materials. So this was the subject of many, many, many papers. But that really didn't start a takeoff in the use of viral load for HCV until it was linked to therapy. So back in the early 90s, I mentioned earlier that there was an immune treatment for some viral diseases, and that was alpha interferon. Alpha interferon is a very interesting molecule. It's produced by mammals in general. If a cell is infected with a virus, they tend to produce the alpha interferon as part of what's referred to as the innate immune response that many of you probably are aware of. And the impact of that is to, firstly, decrease the replication of the virus itself, increase the production of cytokines, increase the production of natural killer cells, and increase the antigen presentation for immune response. You may recall that in SARS-CoV-2, alpha interferon is a significant part of the innate immune response. And I find it very interesting that bats who are basically immune to these viruses but are infected with them have enormous amounts of alpha interferon in their blood and in their cells. So I find that very interesting. I'm jealous of the bats. I wish we had more alpha interferon. Very early in the 90s, there were papers on the attempts to use alpha interferon in treatment of non-A, non-B hepatitis, or later hepatitis C. But there weren't any particularly good measures of the response other than clinical response. So there was a great deal of interest that perhaps we could use HCV viral load in that capacity. That began a long collaboration with a fellow named Johnson Lau, who was at the University of Florida at the time, who became a great friend. I still talk to him once in a while. He was a great collaborator. And he was in close contact with a woman named Jan Albrecht, who was the head of clinical studies for interferon at Schering-Plough. And we struck up a relationship and started doing studies. There were a number of papers that started in 1994 and continued on for many years after that. I'm just going to try to give you a sense for what we learned and how viral load was used as a result. So Johnson Lau, Gary Davis, who we work very closely with, Bob Perillo in New Orleans, a number of other people around the world, they all had really great sample banks that we were able to use to make a number of different discoveries. For instance, we were able to look at people that had chronic hepatitis C infections and found that for most of them, their viral load didn't change very much over time. It stayed fairly flat, although over time it appeared as though they started to go up. But there was another group of people that was very unusual in that they had incredibly fluctuating concentrations of virus. Every time you took the sample, it might even be undetectable and then back to several million copies per mil again. Very odd. Never really did find out why that was the case, but that was observed in a number of people. We found that when these individuals were treated with interferon, there were a number of different response types. One was what you would hope for. Over the course of a few weeks, the viral load decreased dramatically. In fact, in those people that were successfully treated, it became undetectable in their blood. And that correlated well with the clinical outcomes as well, and with other markers such as ALT and AST. But now we saw that this was having an actual impact on the amount of virus in blood, and therefore probably reflective of whatever reservoirs the hepatitis C was being produced in. We found there were people that had a mild response. It didn't go down that very far. And once the drug was taken away, it came back up to where it was before, much like we saw in HIV. And there were people that didn't respond at all to alpha interferon. And at the time, it was very unclear why. We were looking at correlates of that. What could we say about the difference between people who do and don't respond? Well, I mentioned last time we did see differences in genotype. Genotype 1 was more difficult to treat, evidently, than 2 and 3. Um, but when we really looked at it more carefully, the one major predictor for the likelihood of response was viral load itself. And what we found was that at the level of about 2 million copies per mil, and at the time people call it equivalents per mil, equivalents to the virus, we found that if people had more than 2 million mEq per mil of their blood, they in fact almost never responded. People that were below this 2 mEq, 2 million equivalents, people that were below that were more likely to respond, sometimes as much as 70 or 80% of the population, but very low above that. We did studies all over the world along this line because all the experts will want to find out, does it work the same in my population? Does it work the same in my population? Everybody wanted to do that. They wanted to see that it actually responded in their population the same as it did all the other places we looked at. We got to a point where the evidence that was mounted was very compelling. We had our partners in Japan, Daiichi Pure Chemicals, that was responsible for commercialization of our products there. And we were working closely with them because in Japan, there was a very high incidence of hepatitis C infection. So they were very concerned about all this. And as a matter of fact, they were using alpha interferon to treat people. They were spending $2 billion a year on treatment of individuals with HCV infection with interferon. Now, they had the equivalent of the FDA there that was called the COSATIO at the time. And the COSATIO was responsible for this cost. And they were also responsible for approval of the drugs. Interferon was already approved. And for diagnostics, no viral load test was approved. We talked to them quite extensively about what we were doing, and they were very interested in this. And that compelled us to work with the people at Daiichi to do a series of clinical studies. And at the time, I thought maybe we need to do one or two. Our colleagues at Daiichi Pure Chemicals did 10 separate studies throughout Japan. And what was really amazing was that not one person in any of those 10 studies above 2 mEq responded to the interferon. Not one. And there were a number of people, of course, below. So they actually set up a cutoff. They said, this point going forward, COSATIO was going to accept that if you're below that level, you can be treated. If not, and you're above, you're a candidate for drug trials that are going on, and hopefully something will come along, but you're simply just not going to respond to the interferon. Later on, ribavirin was added to the interferon, and that improved it somewhat, but there was this pretty clear cutoff. After we did the studies in Japan and presented them to the COSATIO, they gave Chiron and Daiichi the fastest approval of a diagnostic in the history of Japan, 4 months. That was amazing. So they recognized the importance of it. It was taken up by all the major laboratories in Japan and made generally available. So with this in hand, we went back to the people at Schering-Plough in the United States. And we also then opened up communication with Schering-Plough in Asia. Now in Asia, they were really very excited about working with us. In fact, they conducted a number of campaigns for us, advertising campaigns, explaining to physicians this issue of mEq, 2 mEq. And there were pictures of this, many people from Schering-Plough out talking about it, because they recognized that in many cases, people were not using their drug because they just didn't get a good response rate. Now they could improve that dramatically by testing first, and then using it to actually monitor whether it's effective or not. The people from Schering-Plough in Asia recognized the fact that it was likely that their drug was going to be used more often now because it was easy to tell who would or wouldn't respond. So that made a big difference. But when we talked to the people in the United States, we talked to our old friend Jen Albrecht, and she thought it was a good idea to be doing the testing. But the people in charge of the commercial organization thought it was a terrible idea. They thought they were going to sell less drug. And in fact, as time went on, I think it was pretty clear that the use of the viral load testing to determine who should or shouldn't be treated or to monitor them, that increased the sale of the drugs. So that was what was done all the way through until you see the advent of these direct acting antivirals, the DAAs. So I had mentioned earlier that the ability to culture the virus didn't really occur until 2005. First reported case of a clone version of HCV being able to be cultured in cells. That led to a number of other innovations there, including Charlie Rice's work, which led to the Nobel Prize. But what the most important thing about that is that people could start to look for direct acting drugs in tissue culture first, in cell culture first, and then translate that into compounds that are likely to work in humans. That went very quickly. We started to see these DAAs become available generally from a number of different companies. And as time went on, these direct acting drugs became so effective that nearly everybody was cured. I mean, truly cured of the infection. And what did we see? The result of that? Viral load goes to undetectable, doesn't come back in almost every person. So now there's an expectation that most everyone is going to respond to these drugs. It's no longer dependent on a genotype, no longer dependent on a viral load. So once the virus is cleared, you're clear. That's changed the use of viral load. It's not so much about quantity now because we don't really need to know what the amount is at the beginning or during the treatment. We just need to know that it's cleared. But qualitative RNA detection is still very important because uh, what is done in trying to diagnose someone is that you use the antibody first, antibody test, and you show that they have been or they are infected with HCV. You don't know which it is. You don't know if it's active or cleared. So that is the determination of that. What's the next step? You do a viral load test, a virus test. You determine whether someone has the RNA. It's detectable in the blood. It's an active infection. That is then the trigger of treatment. So that is still typical today, and it will continue to be so for quite a long time. There's been great advancements in the overall treatment of hepatitis C. I think the best example that we are aware of is work that was done in Egypt. Egypt has had a lot of hepatitis C, which is traced back to a treatment program for schistosomiasis using needles that evidently weren't quite as sterile as they should have been, and infected a large number of people within Egypt. The government decided they are going to do something about that, and they went out and they made deals with Gilead to get access to the DAAs. They have antibody testing, they have viral load testing, and they actually have a package which costs less than $80, and that involves diagnosis and treatment for a cure. $80. The company in Egypt that put this all together is called Farco. It's a pharmaceutical company, and they're now looking at being able to sell that package to other people that want access to this. Unfortunately, not in the United States. In the United States, we still have remarkably expensive DAA, and I hope that gets dealt with in the not too distant future. One of the biggest remaining problems in hepatitis C is the fact that it's so difficult to find the people who are infected. We know there are IV drug users who have a much higher likelihood of developing hepatitis C infection, other people that have had blood products that weren't properly tested. But there are still a great many people out there that are not diagnosed. It's still estimated by the WHO there are at least 58 million people worldwide infected with hepatitis C. And we're a long way from eradicating the disease. We need to have access to more of the DAAs. We need to have access to the diagnostics more broadly. And it's not going to be something where every single person is going to be tested. I don't think that's very likely. I know there's a push in some places to do that, but it just doesn't seem economically feasible. On the other hand, targeting people that are at higher risk is attractive and still very difficult. Also, it's not likely that availability of diagnostics and DAAs is going to be sufficient. We're almost certainly going to need to have vaccines for prevention of hepatitis C infection in the first place. And We talked a lot about that with our old friend Mike Houghton from Chiron on our podcast, Halteris Presents, talking about hepatitis C and his great involvement in it, but also the work he's doing today in vaccine development and the importance of that. So this is the last of our 3 episodes on hepatitis C, on interesting stories in the history of diagnostics. And I think it's been something I've been very interested in, obviously. Don't normally devote this much time to one subject. It's just such an interesting story. That's why it's an interesting stories in the history of diagnostics. And I think we've made incredible progress over the years, figuring out what it is, figuring out how to diagnose it, developing drugs against it. And the last piece of the puzzle is really vaccines. And then it's a matter of how to put all that together and find ways to prevent and treat so effectively that we actually eliminate it from all the countries and finally eradicate it from the planet. It should be possible to do that. It's only infecting human beings as far as we can tell. Therefore, if you take it away from the human population, there are no reservoirs left. We should be able to eradicate it, but it's going to take the will and the money from a number of places in order to make that all possible. So thank you once again for watching another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteris Associates. Really appreciate your watching. Bye-bye.